In the second lecture on introductory brain concepts, we're going to look at the protection of the brain via the meninges and the arterial and venous circulation of the brain. That's going to be important to understand when we look at things like stroke and how a stroke can affect different parts of the brain. And then we'll discuss cerebral spinal fluid, which flows through two layers of the meninges. So the brain and spinal cord are part of the central nervous system, and one of the distinguishing features of the CNS, or central nervous system, is that it walls itself off in a way from the rest of the body, and one of the ways it does that is through the cranial meninges. So this is really three layers of membranes that separate that kind of line the central nervous system and separate it from the body. Really, it helps to protect the CNS. It also provides a pathway for the cerebral spinal fluid, which gives buoyancy to the brain. And I mentioned that the brain itself is heavy enough to crush the delicate blood vessels at its base if it weren't for the CSF keeping it buoyant. So let's look at these meninges. There's three layers. The first is the outermost layer, and that's called the Dura Mater. It's a thick, double-layered, durable membrane. It's closest to the skull, so if you just peel the skull off, you'll see the dura there. And it's composed of dense, irregular connective tissue. It contains large blood vessels, both veins and arteries. And these arteries will plunge into the brain and split into capillaries via the pia mater and then go deeper. The cranial dura really consists of two layers. Spinal dura, that's what's around the spinal cord, is only one layer. There is what's called the periosteal layer, which is the external layer, and the meningeal layer, which is internal. That's up against the next layer of the meninges. These layers are really fused, except in places where there are big gaps that open up to form what are called the dural venous sinuses. So I'll show you pictures of the venous sinuses in the next slide. You can kind of see one here to the right. So you can see on the very out, here's the skull on the outside. Let me do this in red. So skull on the outside, here's the brain underneath. And then the meninges are really this layer right here. So everything in between here is meninges. So what's in kind of white, that is the dura. And you can see right here the dura actually splits and there's this gap. You can only see half of it. And this is actually the where venous blood will collect. So as all the blood drains out of the brain, it's going to collect in these spaces. And so that's found in the dura. So those are called the dural venous sinuses, and they all drain into the internal jugular veins, which then brings it back to the heart. There are three extensions of dura that actually separate parts of the brain, and we'll look at these in more detail. So the falx cerebri separates the two hemispheres of the cerebrum, the outer portion of the brain. The falx cerebelli separates the cerebellum into two hemispheres, and then what's called the tentorium cerebelli separates the cerebrum from the cerebellum. So we're going to see in neurology some disorders are labeled as supratentorial, others are subtentorial. Supratentorial means it's above the tentorium. In other words, it's probably in the cerebrum, versus subtentorial is going to be in the cerebellum or maybe brainstem. All right, so that's the dura and the subdural spaces. One thing that can happen is usually this happens from a head injury, is that the small veins that connect the next layer of the meninges, the arachnoid and the dura, can tear, and this forms a bleed within the between the dura and the arachnoid called a subdural hematoma. So subdural hematomas usually a result of head injury. You can also get an epidural hematoma that's on top of the dura. The problem with those is that as they bleed, the blood begins to fill the dura space, and this bleed gets bigger, the pool of blood gets bigger. It can't expand outward because of the skull, so what it does is it compresses the brain. And so this is one of the things we're always looking for after head injuries, and we'll discuss this in detail in neurology, is did a subdural hematoma develop? And sometimes it takes 12 hours, 24 hours or longer for this to become large enough to significantly impact brain function. So we're always really cautious the first day or two after a major head injury, watching out for any change in neurological function, which could be a sign of a subdural hematoma. usually the main thing that's done emergently to sort of help with that would be to actually drill a hole through the skull to relieve that pressure or remove a portion of the skull to do that. All right, so that's the dura. The next layer in is the arachnoid mater that has sort of a spider-like appearance. It's thin, it's avascular, meaning no blood vessels, and then blood vessels pass through it, but in and of itself, there's no vessels that supply it. And then there's cells and loose connective tissue, and there's a lot of elastin, which makes it stretchy. There's a space underneath the arachnoid, between the arachnoid and the final layer of the meninges, called the pia, called the subarachnoid space, and that is where your cerebral spinal fluid flows. So here in the upper diagram, we see the dura mater, and then the arachnoid in green here, and then pia is the deepest one, and between the pia and the arachnoid is where the cerebral spinal fluid flows. If you look over at this picture, you can see more clearly the dural venous space here. And then here the arachnoid is it's mostly white with these kind of green little extensions like spider webs. And again, that's where the cerebral spinal fluid flows. And there are blood vessels, veins and arteries that go through, but the tissue itself is largely avascular. It doesn't have its own circulatory supply. And then the innermost layer, which is going to be actually touching the brain tissue, is going to be the pia mater, and that's depicted as this thin layer right here. So that's pia. This is all arachnoid in here. Actually, arachnoid technically is this layer right here, and then this is the space between the arachnoid and the pia, and that's where CSF circulates. And then we have dura mater, this very thick connective tissue on the outside. All right, so that gives you a little overview of the meninges there. Now, just like you can get a subdural hematoma, you can get a hemorrhage in the subarachnoid space, and that's typically either from trauma or that can be from an aneurysm that's been there, for years and it bursts, and that leads to bleeding. Blood fills up in the arachnoid space, and again, the same thing here is it'll start to compress underlying brain structure, and kind of like a stroke will affect the blood supply to different parts of the brain. And then again, the pia mater is the very delicate inner layer, firmly attaches to the surface of the brain. And again, this is also in the spinal cord. It's thin squamous to cuboidal cells. There's some collagen, again, some elastin. It's pierced by blood vessels as they go in and out of the brain. So in particular, capillaries kind of from this region enter the brain and supply the outer regions of the brain in particular. And then larger blood vessels will go deeper. And then there are denticulate ligaments, which are thickenings of the pia mater, which extend into the arachnoid. So in this particular picture, see a little bit of, we see these little indentations here. They go down into the brain. These are actually following the deep grooves in the brain. Again, this is all pia mater. And then there's some pia mater that actually comes up a little bit into the arachnoid space. All right, so that's the meninges. And again, we're just focusing on the brain. We'll see the same thing when it comes to the spinal cord. Now let's look a little at brain circulation. And again, this will be really important in the pathology section. So the adult brain is about 2% of body weight, consumes, though, about 20% of all the oxygen and glucose used in the body. Interestingly, the only other organ that takes up this much oxygen and energy is the kidneys. So we'll be seeing there's a very strong relationship between the kidneys, which curiously have a bilateral symmetry just like the brain, which is very unusual for an organ below the diaphragm. All the other organs there, all the digestive organs, are asymmetrical. The kidneys retain their symmetry. And embryologically, interestingly, the kidneys begin as a structure in the throat region, the neck region of the embryo, so towards the brain, and they migrate downwards to their adult position. So curiously, we can think of the kidneys as having a connection in that way, morphologically, with the brain. Embryologically, they come from different tissues. Kidneys come from the mesoderm, brains from the ectoderm, but they retain this kind of relationship, which we'll explore in detail when we look at the kidneys. All right, neurons use glucose almost exclusively for ATP synthesis, so your muscles and whatnot can use other things like ketone bodies, which are the breakdown ketones from fat, but the brain needs glucose. No glucose is stored in the brain. There's no glycogen, which is a storage form of glucose. That's only in liver and muscle. So the liver in particular has to supply the brain via the blood vessels the glucose that it needs. If there's low glucose and poor circulation, we start to see things, neurological symptoms like dizziness and confusion, and then potentially convulsions and even worse. If there's a disruption in blood supply to the brain, in 1 to 2 minutes, this will impair neural function. In 4 minutes, we have permanent injury. And this is why in things like CPR, they really emphasize getting the chest compressions going. Chest compressions in CPR are only about 30% effective they only put out about 30% the normal cardiac output that the heart normally puts out. So you're not getting quite as good perfusion of the brain, even with well-performed CPR. But if it's started early and initiated, then you're at least getting some blood supply to the brain and getting some oxygenation to the brain cells. So 4 minutes is when we see permanent injury of neurons in the brain. So how does blood get to the brain? Well, it gets there first through two major arterial systems. So we have the carotid arteries, and they branch off. So if we, not kind of showing, this is only, I'll start with the second diagram down here. If we look at, the blood vessels coming off the heart, the carotid arteries branch off, and the carotid arteries actually branch into an internal branch and an external branch. So the external carotids right here. Here we have one here and one here. And then the internal carotids here and here are the two branches of the carotid arteries. The external carotids go to supply the face and the scalp. So they're only going to stay superficial, and they're going to supply the scalp and your facial skin and muscles and whatnot. Versus the internal carotids will branch and go into the brain. And they actually join up. If you look at the internal carotid here, maybe I'll draw it in black, make it easier. Internal carotid here joins up with the other internal carotid in this little region here. And this is kind of not easy to see in this picture, but this is actually occurring at the base of the brain right here. This is a region called the circle of Willis. So both internal carotids feed into the circle of Willis. And then there's another arterial system that supplies the brain, and that is the vertebral arteries. So the internal carotids are going to supply about 3/5 of the cerebrum with blood, but the vertebral arteries supply the rest. And they travel along the spinal column, so they're actually going to be going through the little holes in the vertebrae on either side of the spinal cord. And they're going to go up through these little foramina, and they're going to enter into the brain. And they're also going to join together to form, you see them here, vertebral arteries. They're going to join together to form something called the basilar artery, which is going to supply a lot of the brain stem, and then that joins into the circle of Willis. And so that's going to supply the posterior 2/5 of the cerebrum, the cerebellum, and the brainstem. Again, where this is going to be important is if you get a blockage of blood flow, and the most common reason for that would be what's called a thrombus. A thrombus is a clot that forms inside of the circulatory system. When we say blood clot, that's the same thing as a thrombus. The medical name, though, we use is thrombus. So if there's a thrombus that can break off and embolize, so when a thrombus starts to form on the wall of a blood vessel, and then it breaks off and travels downstream, as the blood vessels get narrower and narrower, the thrombus can lodge into a blood vessel, and that will disrupt circulation to a portion of the brain. Now that's one way circulation can be compromised to the part of the brain. You can also have a hemorrhage of one of these vessels, and then that's going to cause a loss of blood supply to part of the brain. And we'll discuss all of these causes and how it happens and how we diagnose it in the neurology section on stroke. But knowing where it's occurring and which blood vessel can tell us what part of the brain is going to be affected. So we're going to be looking at the major vessels and understanding what parts of the brain they perfuse. So again, the internal carotids and the vertebral arteries collect at the circle of Willis, and this is at the bottom of the brain. And there are branches that come off the circle of Willis they're going to supply the bulk of the brain. So here is a picture of the circle of Willis. Here's the basilar artery, and these are, again, the vertebral arteries that fuse there. And then we have the carotids that come in, and they're feeding the circle of Willis. And then we have three major branches that come off. One on the front, there's actually two of them, and then we have a middle one, and then there's a posterior one. And this is going to be, these are the major cerebral arteries. So that would be the anterior cerebral artery, the middle cerebral artery, and the posterior cerebral artery. So we're going to be looking at those in detail because, again, they are supplying specific regions of the brain, particularly of the cerebrum, where your higher cognitive functions happen. So if you have disruption of blood flow in any of these, it's going to affect very specific areas of cognitive function. Here's another picture of the circle of Willis at the base of the brain. You see it sort of wraps around. Here is the spinal cord right here coming in. Here's the actual here's the olfactory bulbs, and then cut here are the optic nerves that are coming in. And this is the pituitary gland right here. So it kind of wraps around this area here. Here we see the basilar arteries coming in and going down into Willis. Okay, so, and then from there, the anterior, middle, posterior cerebral arteries branch off, and they're going to supply those major parts of the cerebrum. So that is an overview of the brain circulation. Now, one thing, once the blood is used in the brain, it has to drain, and that's going to be via the veins in the brain, which drain into those dural venous sinuses. And that's going to drain down, and I don't have a picture of that here, into your internal jugular vein, which will then join up with the superior vena cava, and that'll go into the right side of the heart, back into circulation. So that's the venous drainage. Now, importantly, the blood flow to the brain, there's a second layer of protection to prevent a lot of substances from the blood from entering the neural tissues, and that is called the blood-brain barrier. And we already looked at this in the case of astrocytes. So astrocytes help form the blood-brain barrier. And essentially what this is if you look at a capillary, it's essentially just a single layer of endothelial cells. So if you look at a brain capillary or anywhere in the body, and there's one thing unique about the brain capillaries. They have tight junctions between each of them, and that's going to prevent a lot of substances, a lot of polar compounds like glucose from just moving through into the brain, from the blood to the brain. In addition, there are these astrocytes, and they form these projections around, they sort of wrap around, that's an astrocyte projection. Maybe here's another one. And they form around, and they're going to form an additional layer. So anything in the blood's going to have to go through two layers to get into the brain. And there are special transporters, and we'll look at some of those, but there are different transporters for glucose and so forth. So glucose has to transport across the blood-brain barrier via active transport. Other substances like creatinine, urea, different ions can cross the blood-brain barrier, but only very slowly. They have to get through. There's different leak channels and whatnot. And then many proteins, many drugs don't cross the blood-brain barrier. Most polar compounds don't readily cross. They have to be transported. So amino acids, for example, you eat a lot of tryptophan. Most of that tryptophan from the diet doesn't get into the brain. In fact, there is an amino acid transporter which is conveying it into the brain at a constant rate. So you're not going to flood the brain with tryptophan if you eat a bunch of it with dinner. However, there is a compound, a metabolite, remember, of tryptophan, 5-hydroxytryptophan. That's the precursor to serotonin. That is actually readily absorbed across the blood-brain barrier. That doesn't compete with that transporter. That amino acid transporter actually is transporting a lot of other amino acids, so it gets really backlogged. But the 5-HTP can get into the brain more quickly, and so it could potentially raise serotonin levels and so forth. Lipids and lipid-soluble substances do diffuse across the blood-brain barrier without major problems. So fatty soluble things, and I mentioned curcuminoids in turmeric, are actually lipophilic. So they can get through the blood-brain barrier, and people that eat a lot of curry all their life actually have brains that look orange from all the curcuminoids that have accumulated there. So those are some examples. Now we think that trauma, for example, head trauma, certain toxins, inflammation, all these can break down the blood-brain barrier, making essentially a leaky blood-brain barrier. And there is some speculation that that actually, and some good data actually, that that causes a whole host of different neurological conditions, from just simple confusion and what we call brain fog to all sorts of other issues. So that's something that a lot of more functional neurologists will look at is, is there evidence of leakiness in the blood-brain barrier? And is that letting things pass into the brain that shouldn't normally? And is that creating issues? So there's some nutritional things that are done for that and so forth. We can discuss that later. So that's a little on the brain blood supply. We're going to be coming back to this again in more detail when we look at stroke in the neurology section. Finally here, let's jump back to the meninges and look at the cerebral spinal fluid. This is a clear, colorless fluid. It's composed mostly of water, and it protects the brain and spinal cord. It has a volume of just under 150 milliliters, so not a large amount, but it's under constant and steady pressure, and it circulates continuously throughout the brain and spinal cord in that subarachnoid space. It carries oxygen and glucose, some waste products like lactic acid, urea, different ions, and then some white blood cells, although normally in a healthy state without infection there shouldn't be a lot of white cells. The functions of CSF is number one, mechanical protection, just giving a extra cushioning between the central nervous system and the body. There's a homeostatic function, so the pH of the cerebral spinal fluid is going to be affected by pulmonary ventilation, and that's going to affect cerebral blood flow. We actually know, interestingly, that when you breathe, that actually pumps the CSF up towards the head, and then when you inhale and when you exhale, it goes the other way. So there's a pumping action of respiration. And we actually now have evidence that people that breathe in through their nose versus their mouth, so nose breathers on the inhalation versus mouth breathing, actually have better performance on memory tests and other types of functions. So curiously, mouth breathing versus nose breathing has different consequences on the cerebral spinal fluid and how it's flowing, and that can affect brain activity. So that was a recent finding. And then there's a circulation exchange of nutrients and waste products to the CSF. In the intro neurology section, I mentioned at night how the neurons, normally neural tissue and glial tissue, is usually very dense with very little water and very little space between the cells. But at night, the neurons actually contract a bit, and that opens up extracellular space around them, and CSF can wash through that and wash away all the waste products and whatnot that have accumulated during the day. And so that's yet another benefit here of sleep and healthy sleep rhythms. So the CSF actually flows, again, around the meninges. It's actually produced within the ventricles. So if you look inside the brain, again, I didn't show you pictures of this earlier, but if you look at the cerebrum, here in kind of the bluish color, purple, we see these kind of big structures, and these are hollow spaces called the ventricles. And there are actually 4 of them. So here's one right here, here's 2 behind it, and this is looking sideways here, and then here's the 3rd ventricle, which is more midline, and then here is the 4th ventricle down here by the cerebellum. So cerebral spinal fluid is going to actually be formed in the 1st and 2nd ventricle. It's going to flow through this little duct down into the 3rd ventricle, and then it's going to flow through another duct called the cerebral aqueduct down into the 4th ventricle, and that's going to flow down into around the spinal cord and so forth. And then it's all going to come back up the spinal cord. So if you look at this second picture, shows you a little bit better. So here you see these little red structures at the 1st ventricle. Some of them are in the 3rd ventricle. These are called the choroid plexuses. And the choroid plexus is what secretes cerebral spinal fluid. And so it secretes it into these spaces. It's going to flow down 3rd ventricle, down into the 4th ventricle, and that's located again between the cerebellum and the midbrain, right here in the brainstem. And then it's going to go down around the spinal cord, all the way down, and then back up again. And then it's going to flow around the pituitary. It's going to flow all the way around, and eventually it's going to drain into, and you see here, these little arrows here, these little projections that go up into the dural spaces. It's going to drain into the venous spaces. and that's going to then drain into the internal jugular vein and go back to circulation. So that is kind of the circulation of the cerebral spinal fluid. I showed you kind of the forward pathway. There's also a backward pathway here, so it also goes around the cerebellum and then up and then into the venous spaces, draining there. So these little connections between the cerebral spinal fluid flowing and the arachnoid space and the dural sinuses are called arachnoid villi. So an arachnoid villus is where the CSF drains into the venous spaces in the dura. So that's the pathway of CSF circulation. Again, that's going to be influenced by your breathing rhythm. So we have this independent regulator there, and that really connects the breathing rhythm with cerebral function and what's going on there in the nervous system. All right, so that's the major pathway there. The choroid plexuses, going back up to that, which secrete the cerebral spinal fluid, they have a very specific cell type. This is that fourth type of glial cell we talked about called ependymal cells. So ependymal cells are neuroglia with very tight junctions between them, and they secrete the CSF, and they also absorb waste from the CSF. And so they form what we call the blood cerebral spinal fluid barrier. So those are the ependymal cells in the choroid plexus. Again, you see that in the red down here in this picture. And they're found in the lateral ventricles and some of them in the third ventricle. So lateral ventricles, these are the largest. There's one in each cerebral hemisphere. There is what's called the septum pellucidum, which is a membrane that separates the two. And then CSF is formed in these lateral ventricles and then enters down the third ventricle via these very narrow openings called the interventricular foramina. So that's what connects the lateral ventricles with the third ventricle. Then the third ventricle is midline. It's a single slit-like ventricle. It's actually between a structure called the thalamus, which we'll look at. So the thalamus wraps on either side of the third ventricle. And then the choroid plexus and the third ventricle add additional CSF. And then that's going to flow via the cerebral aqueduct into the fourth ventricle, again down here by the cerebellum. And then the fourth ventricle has no, it doesn't add more CSF. It actually just is continuous down here with the central canal of the spinal cord as well as the meninges around the spinal cord. And so that's going to allow the CSF to flow into the subarachnoid space, and that's going to circulate around the meninges as we've talked about. Okay, so that is the basic pathway of cerebral spinal fluid. Now again, we'll be touching back on this as we look at number one, different CSF disorders. So for example, if that CSF doesn't drain properly from the ventricles, let's say there's a blockage in the cerebral aqueduct down here, going back up to this picture, CSF will build up, pressure will build up in the third ventricle and the first and second, the lateral ventricles, and that can create something called hydrocephalus. So hydrocephalus is when the CSF, the normal drainage routes are blocked somewhere. And that can be due to a tumor, for example, but that can also be due to structural problems. There can be congenital reasons for that. There's a lot of reasons for hydrocephalus. But that would typically cause, increased intracranial pressure. So as that CSF builds up, it actually puts pressure on the underlying neural tissues, and that can affect function. So that can create headaches, that can create visual disturbances like double vision, things like that. It's a very sensitive area, as we'll see here in a couple of slides. Down here, or the next couple of little lecture sections, down here in the fourth ventricle, up against the brain stem right here, there's an area that's actually called the area Pastrima. And this is one of our vomiting centers. And so the area Pastrima is sensing what's in the CSF, and if there are things there it doesn't like, it's going to trigger vomiting. And that's sort of a natural protection to try to get out of your digestive tract, for example, whatever might be being absorbed into your system. And so that can also increase intracranial pressure, can trigger that vomiting center, and people can just start uncontrollably vomiting. So again, we'll look at that with hydrocephalus, but that's one potential pathology that can occur here. We'll also come back to this from a more, say, esoteric perspective. If you kind of follow the outlines of the CSF of the ventricles here, the first, second, and third ventricles, and if you look at the regions of the brain, so I mentioned the thalamus is right here, and the third ventricle is sandwiched between them. Right up here we have the hypothalamus pituitary, and then back here the pineal gland. If we look at all these different, I guess a little higher pineal right there, if we look at these regions and take sort of a plaster mold of them, you actually get this shape which in ancient Egypt was known as the Eye of Horus. And interestingly, this was associated with the old mystery centers and initiation centers. And the idea was that when you do special inner exercises, you can open up these structures of the brain and start having extrasensory perception and start to perceive things in different dimensions. So part of this, these areas of the brain might be involved in developing higher inner vision. We do know these parts of the brain are important for example, forming imaginations, inner pictures. So our imagining capacity and so forth is tied in neurologically with these brain centers. So we'll come back to it from that perspective as well. All right, so that summarizes a little bit here on the meninges, the cerebral spinal fluid, and the cerebral circulation. And in the next lecture, we'll start in and look at the brainstem and all of its corresponding structures.